Hey guys, I hope you're doing well today. I bring you part 8 of Unit 4, and this will be somewhat of a departure from our previous 7 lessons in this unit because today we're not talking about cell division, mitosis, or meiosis, but rather we're talking about cell-to-cell communication, how cells communicate within a multicellular organism like yourself. And specifically, we're talking about a particular type of cell-to-cell communication known as a signal transduction pathway. We're going to be covering pages 114 to 119 in the Campbell Biology in Focus textbook. This topic, signal transduction pathways, has become sort of a darling of the AP biology community. The College Board, which is the company that creates and administers and grades the AP exam, they really have latched on to this topic in recent years. They love to ask about it especially on free response questions, and so it's pretty important. And so we're going to spend some time today. This lesson is actually going to be a fairly brief, at least I know I always say that, and it's usually not true, but I'm going to try to keep it fairly brief. And this will serve as an introduction to another lesson that I'll be posting, sort of a companion piece, and you'll need to watch both lessons. This is going to be a general overview of what signal transduction pathways are all about, and some of the vocabulary associated with them. And then I've got another lesson by another teacher. It won't be me, but somebody that I trust a lot, and I'm going to share another lesson specifically about one great example of how a signal transduction pathway works. And so I'll post both lessons at the same time. You'll need to watch this lesson first, and then watch the second lesson, and what I'll do is I'll give you a quiz that covers both lessons together. So that's our plan. Let's go ahead and get started. All right. So I've always sort of imagined signal transduction pathways, STPs, as being like a Rube Goldberg machine. And if you are wondering what the heck I'm talking about, you've probably seen a picture of a Rube Goldberg machine before. Maybe you didn't know what it was, but Rube Goldberg was an American cartoonist who would design these extremely intricate, unnecessarily complicated machines to perform very simple, mundane tasks. And it was all sort of for the purpose of getting a laugh and sort of entertaining folks. But you can see you're looking at an example of a Rube Goldberg machine. This gentleman is eating soup, and he needs to wipe his mustache with a napkin. And so when he lifts the spoon to eat the soup, that pulls on this string which actuates a lever on this spoon and causes the cracker to flip up in the air. The parrot grabs the cracker. That causes this mechanism to spin around and dump something into a bucket, which pulls on another string, which causes a match to light, which fires the rocket, which pulls this string, and the napkin swings across his mustache. And it's comically complicated. And I don't mean to suggest that signal transduction pathways are unnecessarily complicated, but they have lots of steps. And you'll find that with a signal transduction pathway, there's usually some fairly simple signal that arrives at the cell. But that simple signal, which in this case, is a man raising a spoon, kind of sets in motion a whole series of events. And at the end of that domino effect, the desired action takes place. So it's somewhat like a Rube Goldberg machine. A signal arrives at the cell, a whole chain reaction of multiple steps takes place, and then at the end, something happens that is a desirable result. Either an enzyme becomes activated, or maybe the expression of a gene gets altered, but there's multiple steps that have to happen in between. So let's see how this works. Okay, so here's just a definition. I know this is kind of a wordy and lengthy definition, but a signal transduction pathway is a set of chemical reactions in a cell that occurs when a molecule, such as a hormone, some kind of chemical signal, attaches to a receptor. And that receptor can either be on the cell membrane or it can be inside the cell. And we'll talk about why that is. It's important to know the position of the receptor, and we'll talk about that. Once that chemical signal is received by the receptor, either on the cell membrane or inside the cell, the pathway begins, and there's a cascade, a domino effect of biochemical reactions. This is where it kind of gets like a Rube Goldberg machine, because one thing happens which causes another thing to happen, which causes another thing to happen, on and on and on, a whole series of events. We call that a cascade, until eventually the signal or the cascade reaches the target molecule or a target chemical reaction. Thus, the pathway is a method by which molecules inside the cell can be altered by molecules on the outside. So, wow, that's a lot. And I don't think you fully can appreciate this lengthy definition until you see an example. And we're going to look at a very specific example of a signal transduction pathway, one that's very important in your body, one that plays a big role in your fight or flight response, which is how you deal with frightening or life-threatening or stressful situations. And we're going to look at that very specific example of a signal transduction pathway in a separate lesson. So let's continue now with the introduction to signal transduction pathways. All signal transduction pathways begin with a signal. So there has to be some molecule released by a cell that travels to a cell, and then there's going to be a receptor, a protein, that's going to receive that message. So in this picture, the little blue, I guess those are blue, the tiny little dots, those are the signals. Those are the molecules that act like signals. And see, there's going to be a receptor right here that receives one of those signal molecules, and that's going to trigger the cascade. That's going to push that first domino, which is going to cause all the other dominoes to fall. Now, if a cell releases a signal and it stimulates that same cell, so if we're talking about the cell releasing a signal to stimulate itself or to communicate something to itself, that's called autocrine. That's an autocrine signal. The signal affects the same cell that released the signal. And a couple of examples, just in normal healthy cells, a lot of the chemical signals that trigger cell division are autocrine signals. So the cell will produce a protein that binds to a receptor and then triggers the beginning of mitosis. In tumor cells, this process starts happening out of control. So if this is a cell, like a cancer cell that's in the middle of a tumor, it will release signals, massive amounts of signals that will cause that cell to divide way more often than it should. And so you get this out of control cell division that causes the tumor to grow and grow, and that's sort of the bad part about cancer. So autocrine is a cell communicating with itself. You can also have paracrine signals, which basically it's a signal that's communicating to a nearby cell. So maybe you've got two nerve cells, and this nerve cell needs to communicate with another nearby nerve cell. If you know anything about neurons and the way the nervous system works, there's always a tiny little space in between any two nerve cells in your spinal cord, in your brain. There's always a little space in between. They don't actually touch each other. That little space in between the two nerve cells is called a synapse. And the chemicals that travel across the synapse from one nerve cell to another are called neurotransmitters. And this is how your nerves work. This is how neurons talk to each other, and it's how nerve signals, what we call action potentials, get transferred from one neuron to the next. So we'll actually, later in the course, we'll talk more about this, but neurotransmitters traveling from one neuron or nerve cell to the next is a great example of paracrine signaling. We're going to focus mainly today, certainly our specific example that we're going to talk about is going to be endocrine. Endocrine signals are long distance signals. So this is where a cell in a gland, this could be in your pituitary gland, it could be in your adrenal glands or your pancreas or any of those glands. The gland cell releases a signal which doesn't go directly to another cell, but instead the signal molecules diffuse into the bloodstream. They get picked up by a blood vessel. And then the circulatory system, when your heart beats and pumps that blood throughout the body, those signals, which we call hormones, this is what hormones are. Hormones are just signals that travel in the blood, and they travel to a whole nother part of the body. So for example, we're going to talk about a hormone called epinephrine, which is also called adrenaline, by the way, same thing. Epinephrine is released by your adrenal glands, which sit right on top of your kidneys, and it gets the hormone epinephrine gets picked up by the bloodstream, and it travels it travels all over the body, but it travels to the liver, and your liver cells become the target cells. The epinephrine molecules will leave the blood and bind to receptors on the surface of the liver cells. So this is long distance. This is traveling from the adrenal glands to the liver, from one part of the body to another, and these chemical signals travel in the bloodstream. We call those endocrine signals. And you have a whole system of your body that does this. It's called the endocrine system. All of the glands of the body, I named a few of them, the pituitary gland, the thyroid gland, adrenal glands, what am I forgetting? The pancreas. Those are all endocrine glands. And so your extra credit phrase that pays today is this: the endocrine system is all about long distance communication. The endocrine system is all about long distance communication. Okay, so it's important that you remember the 3 stages, the 3 steps that always occur in a signal transduction pathway. They're pretty simple, but very important that you remember: reception, transduction, and response. So the first step is receiving the signal. Reception means receiving the incoming signal. Now, what part of the cell does that? The receptor. So every cell in your body has multiple receptors, and they each are designed to receive a particular incoming signal. Very specific. Each receptor will only bind with one kind of signal molecule. The receptor is a protein which can be located on the outer surface of the cell membrane, or sometimes the receptor is located inside the cell, inside the cytoplasm, and sometimes even inside the nucleus. And so we'll talk about the factors that affect the location of the receptor, but the receptor is the protein that actually receives the incoming signal. That signal molecule has a fancy name. It's called a ligand. Ligand. Lig means to bind. That's where it's the same root word as ligament. You know, your ligaments bind your joints together. So anytime you see Lig, it means to bind or to tie together. Ligatures are pieces of rope that are used to tie hands and feet together. So lig or lig means to bind. And so the chemical signal that binds to the receptor is called the ligand. And a lot of times that's going to be a hormone. The ligand will be a hormone that was released by a cell in a gland, like the adrenal gland, and it has traveled through the bloodstream to some other part of the body, and the target cell is the cell that has the receptor, because the target cell is the cell that receives the incoming message. Okay. The second step is called transduction. This is the really important part. Well, I guess all of these are important, but this is the part of signal transduction pathways that remind me of a Rube Goldberg machine. You can think of the ligand as being like the man holding the spoon. You know, remember in the Rube Goldberg cartoon, the man was holding a spoon. He was eating soup. So the spoon was like the ligand. But transduction is everything that happens after he lifts that spoon. So transduction is that chain reaction that is triggered by the change in the receptor protein, which happens when the ligand binds to it. So once that signal, the ligand, is received by the receptor, that receptor protein actually changes. Usually it'll change shape, or it will something will change about its structure. That would be like the man lifting the spoon. So he's moving its position, he's changing something about the spoon, and that sets in motion a whole series of events where one thing happens which causes another thing to happen inside the cell, which causes another thing to happen and another thing to happen. That chain reaction is the transduction step. And usually there's also a process called amplification where the signal gets amplified at the same time that it's being transduced. And what that means is the magnitude of the signal gets increased. So you might have just one tiny molecule that binds to the receptor. But, you know, that's a tiny little thing. It's one little tiny molecule. But those steps that happen inside the cell trigger a massive response. So we call that amplification. It's like it's sort of like when you play an electric guitar, which I wish I could do. You're causing tiny little vibrations on the guitar strings, but when you run that through an amplifier, it makes this massive sound which can fill stadiums, you know, with sound as it comes through the speaker. So that's the second step. It's called transduction. The third step is the response. This was like the whole point of the signal transduction pathway. This is the thing that needs to happen in the cell. And so this could be any type of very specific cellular response. This could be a gene that gets switched on or switched off. So sometimes the response is either causing a gene to be transcribed and translated, or shutting down the gene, which would be to inhibit the gene expression. Another possibility, and this is a little more in tune with what we're going to be learning about in our specific example, we're going to be talking about the hormone epinephrine, which is produced by the adrenal glands. It travels through the bloodstream to the liver, and when epinephrine binds to a receptor on the surface of the liver cell, it kicks off a chain reaction which causes this enzyme, phosphorylase, to be activated. And when phosphorylase is activated, it begins to break down glycogen, which is a polysaccharide, stored in the liver cell. Now, the cool thing about that is when you break down glycogen, it releases massive amounts of glucose, which is fuel, right? So the whole point of this is you're making fuel available for your cells, and that glucose can go back into the bloodstream and provide energy throughout your body so that you can deal with a stressful situation or run or fight. It's all part of that fight or flight mechanism. So that would be a response, activating an enzyme to carry out a particular chemical reaction. The message has to be received, and then there's a chain reaction that happens inside the cell. We call that transduction. And finally, there's a response. Okay, I mentioned earlier that the target cell, which is the cell here that's receiving the incoming signal, has to have a receptor. There has to be a receptor protein to receive the ligand, the signal. And I told you that the location of that receptor can either be on the surface of the cell, right on the plasma membrane, or it could be somewhere inside the cell. In this case, it's even inside the nucleus. So what is it that determines the location of the receptor? Well, it's simple. It depends on the chemical characteristics of the signal. So if the signal, the ligand, remember this, we can call this a ligand. If the ligand is a protein hormone, like insulin, or if it is an amino acid-based hormone, like epinephrine, that is not going to be able to cross the plasma membrane. Remember, the plasma membrane is a phospholipid bilayer, and it's pretty finicky. It does not allow all materials to easily cross. So molecules that are made out of proteins or individual amino acids are not going to be able to easily cross this barrier. So that means the receptor had better be on the outside surface of the cell, because otherwise there's no way the ligand is going to get to the receptor. But if the ligand is a lipid hormone, like testosterone or estrogen, for example, because this is a lipid and the phospholipid bilayer is also a lipid, then it's going to be very easy for that molecule to get across. So lipid-based ligands, again, testosterone and estrogen would be good examples, can cross the plasma membrane, they can get into the cell, they can even cross through the nuclear envelope, which is also made out of lipid. And so the receptor can actually be inside the cell, and in this case, even inside the nucleus. So here's looking at an example. This is the way epinephrine works. when epinephrine arrives at a liver cell, the receptor, the epinephrine receptor, is located on the outer surface of the cell. When that epinephrine binds to the receptor, it causes this receptor to change shape. That change in shape triggers a series of chemical reactions. One thing happens, causing another thing to happen. This is the Rube Goldberg effect, right? So this is transduction. And then finally, an enzyme, at the very end of this cascade, an enzyme becomes activated and a particular chemical reaction occurs in the cytoplasm. That's the response. Okay. In the example of testosterone or estrogen, that's a lipid-soluble ligand, so it crosses the membrane easily. It enters the cell, and eventually it binds to a receptor inside the cell. And the transduction, in this case, is when this receptor binds to the DNA. It actually binds to a particular gene, and it causes the gene to switch on. That's called, the fancy term is up regulation of the gene. Up regulation means the gene is switched on, transcription occurs, messenger RNA gets made. We're going to talk about that process later. And eventually a protein gets made. So the response would be the building of this protein. But you can't build a protein unless you switch on a gene. And so this was all triggered by a hormone binding to a receptor. The receptor binds to the gene, switches the gene on, and then the response is going to be the synthesis of a protein. Okay. Whew, this is complicated, and the only way you're really going to understand this is to see a specific example. And so that's what we're going to do. And it's going to be a separate video that you're going to watch, but this is just setting the stage. So what you're going to see in the separate lesson is how the epinephrine signal transduction pathway works. Epinephrine is a hormone. It's an amino acid-based hormone made by your adrenal glands. It is an endocrine signal, so that means long distance, right? It travels through the bloodstream from the adrenal glands to, well, all over the body, but to the liver in particular. And when epinephrine arrives at your liver cell, it binds to a receptor. So epinephrine is the ligand. It binds to a receptor. There's a chain of events, a domino effect inside the liver cell that eventually activates an enzyme called glycogen phosphorylase. Glycogen phosphorylase. And what that does, what that enzyme does when it's activated, it breaks down glycogen. Glycogen is a polysaccharide. So when you break it down, you get lots of glucose. All of that glucose goes into the bloodstream and provides fuel for your muscles and your brain and your heart so you can deal with something that's frightening or life-threatening or stressful. That's the burst of energy that you feel when something frightens you, or even just when you're stressed because you have to go make a speech in front of 1,000 people and your heart starts to race. That's that rush of glucose into your bloodstream, and it's all part of this signal transduction pathway. So I'll give you more detail. Actually, it won't be me. It's another teacher that I trust, but I'll be sharing the separate lesson real soon. Make sure you watch this first, then the separate lesson on the epinephrine signal transduction pathway, and you'll be quizzed on both lessons together. And so much for me keeping this brief. I'm just about to hit the 30-minute mark, but hope you have a great day.